We would now like to resume the JIRCAS International Symposium and move on to the second session. Dr. Keiichi Hayashi is the chairperson of this session. Dr. Hayashi, please come up to the stage.
Good afternoon once again. I'm Hayashi, Program Director of Environment Program. So I will chair this session. So this session 2, we will invite distinguished speakers with expertise for major crops. So it is an opportunity for us to learn why genetic resources is so important for those crops to build resilient food system in era of climate change. So first, we have a speaker, Dr. Venuprasad Ramaiah from International Rice Research Institute in the Philippines. He's an established plant geneticist and a rice breeder with experience in Africa, in Asia. He has been in charge of International Rice Gene Bank, where the world's largest rice genetic collection exists, leveraging rice biodiversity to develop climate-resilient varieties. Dr. Ramaiah, floor is yours.
Thank you, Chair, and good afternoon, everyone. It's a pleasure to be here and to see some of the good friends around here, and I would like to thank the organizers for this invitation. So today I've been asked to talk about IRRI strategies to utilize genetic resources in breeding resilient rice. And in the next 20 minutes, I hope I can do justice to this topic. So before we jump into the topic, let me introduce briefly the International Rice Gene Bank. So International Rice Gene Bank is one of the oldest rice gene bank, and it hosts the world's largest collection of rice diversity. We have over 132,000 accessions as of today, which includes a significant number of wild species, about 5,000 accessions. Then this collection is also said to be one of the most diverse in the world because almost all the rice growing countries in the world is represented. And we have almost all the wild species and related genera also represented in the collection. Now, jumping into the topic about how can we use genetic resources for the breeding program. This is the strategy that IRRI is following for breeding. So what are the salient features of this breeding program? This breeding program is targeted at improving the genetic gain for yield for about 1 to 2% every year. That is the main mission of this breeding program. And if the salient features, as you can see, it focuses on elite by elite crosses. Then it uses a rapid generation advance to fast track the development of the varieties. Then it uses genomic selection. Then it focuses on extensive on-farm testing so that we can develop such varieties which have 1 to 2% gain every year. This is the main breeding strategy. And how to bring in other traits is a related strategy where gene bank will be explored. So this is it's in the next slide. So this is the strategy in which by doing pre-breeding, we can bring out the variation or the necessity genes for other related traits other than yield from the gene bank and then supplement the breeding program. So as you can see in the slides on the top portion, you have the gene bank, and we have a strong trait discovery and deployment program which takes care of many of the genetics work, some of which Prakrit already mentioned. So such type of work will be handled, and then those information or those genes or QTLs or haplotypes will be transferred into the breeding line using a line augmentation strategy as mentioned. And the lines, improved lines again goes back into the same breeding pipeline that I showed before. So this is said to be a constant cycle. So this is the strategy to use genetic resources to augment the breeding. So these are some of the traits of interest for the IRRI breeding program based on the market preferences as well as the TPE preferences. So I will not read all of them. This group probably understand this very well, but we have yield-related traits, yield and grain quality I missed, sorry about that. Then there is nutrition-related traits, abiotic stresses-related traits, biotic stresses, and other important traits which are upcoming and important, such as methane emission, retunability, and stuff like that. So these are the traits of interest that we will try to explore from the gene bank. So in summary, IRRI's breeding program currently focuses on quantitative traits such as yield. It will follow genomic selection. But for qualitative traits, for the some which I already mentioned, they use introgressions mainly from the gene bank. And this is the general strategy, which probably is very well established, and many of you are already familiar, which is go to the gene bank, get the material, create genetic specialized stocks of this material, do the phenotype and genotype of them, then do the analysis, then identify the locus of that, and do the introgression. So this is the standard practice across rice or any other crop that we know of. So in terms of specialized stocks, I would like to mention some of that which has been developed and are also still being developed as we speak today. These are the standard biparental mapping populations. There are several dozens of them. No need to go into details of this. It's very common. Then we also have diversity panels. So those in the rice business may know about this. We call the rice diversity panel 1. Then there is 3K panel, which are the sequenced lines. These are used as diversity panels. We have several magic populations targeted at, for different traits. We have the NAM populations. We have the CSSLs, which are for the wild species, to utilize the wild species. So these are continuously being developed, and as we speak, about 24 donors from the AA genomes are being used to develop this. This is going to be a massive resources that could be used for several traits. Then very recently, we also have the eGWAS panels that is being used to map out the underlying genetic factors for heat tolerance. This is in general. So, in this slide, I want to just give you an example of all the kind of QTLs, things are there only for drought that were identified by researchers at IRRI. We cannot go into details of this because of time. Just to give you a sample of, these are some of the QTLs and the underlying candidate genes and haplotypes that the trait discovery program is looking to incorporate into the breeding lines. Then the other examples related to other traits of importance, whether it is nutrition, whether it is stress tolerance, using the magic populations. Then similarly, we have some of the latest results, how CSSL can be used for stress tolerance, whether it is insect resistance or about heat tolerance. So these are different ways in which we are trying to explore and utilize the genetic resources in the gene bank into the breeding program. So this is one main strategy which probably is very well established. Then the other one we are also thinking of is how to create the rice digital gene bank. Because having this type of digital gene bank supports the molecular breeding, not only at IRRI, but in any other center. So basically, we want to sequence, right? The rice community know we have sequenced 3,000 few years ago, and it is a very popular resource. But now the target is, the sequencing is ongoing for the 10,000 varieties. And pretty soon, in a year or so, this will be also publicly available. But the target is to go for 132,000 in the collection, almost sequence everything in the collection and extract the information and to make this information publicly available so that the global community, not just the IRRI breeding program, the global community, especially the rice breeding community, will benefit from this. So this is one other key strategy that we are working on. Coming to the more recent ones, which I am directly leading, is how do we apply artificial intelligence, particularly machine learning programs in gene bank, and that can lead to better utilization of genetic resources. So the strategy which I would say is that we are following is digitize or extract and analyze. So that is what I am calling, this is my terminology. So where are we with this one? So digitizing, especially the seed characters of the entire collection is almost finished. So almost all the 132,000 accessions we have, the seed images have been captured. And now, once the image has been captured, now we are using this as the raw data to kind of extract the information about the collection. So some examples I have given here. This is some are publicly available machine learning models out there using which we can extract the information. And after extracting, we can use this information to do JIRCAS and fast track whatever we have been doing before. So I will give you some examples later. So with this type of raw material, not only we can extract the information and do JIRCAS, we have also developed this model which we call as digital grain identifier, where it can identify any accession in the gene bank based on the seed image. You know, it's quite accurate. Right now it is trained on almost 110,000 of the gene bank collection, and very soon it will be, the whole collection will be trained on it. So this has so many other applications, many of which are related to use. I will come back to that later. So this is one output of that model. So, you know, many times we are we are very proud of our rich collection, but sometimes we don't know what we have and what is the kind of diversity that we have. But with this AI-based tools, we have extracted the information about the whole collection and kind of put it into a biplot here, and it gives you a bird's eye view of how the collection is. This information has also helped us to look, I will come to that again, where we show how this can also be used. Then seed characters was one of the trade, but we are also digitizing the panicle, and before we were using PTRAP, which was developed in Japan, but now we have moved into the machine learning models to extract that. Then we are also looking at the biomass aspect, and we are working with Dr. Tanaka in Yokohama University. And this is an example of how the diversity for biomass there in the gene bank collection. So we have materials about 1 ton per hectare and up to 10 ton per hectare. So this is an important trait which breeders can always use and benefit. How one variety can give so much high biomass than the other one. Then this is work that we are doing with Dr. Takai in JIRCAS, where we are screening the material for retunability and how the technology can be useful. So, for example, you can see the diversity that is available in the collection for retunability, very high to almost low. So we are still in early stages, this is very fairly new work. Now we can extract this information and use this information as a basis to do JIRCAS. So this is the work of our collaborator from Dr. Valier in University of Nebraska in USA, where they have clearly shown that you can extract the information from machine learning models on your panels and then use it JIRCAS, and they have identified a gene called SPED1 in rice. And this SPED1 gene was already reported by somebody else 10 years ago that it is related to panicle characteristics. So it is showing how reliable the new method is, and we can increase the efficiency of utilization of the genetic resources. Coming to the other topic, again, can we use AI and machine learning models to screen for new donors? You know, this is a major challenge which many of the genetic resources people will have, that we have such a big collection, which is important, but when we start going and finding the collection, what is needed for us, it's a nightmare, right? Screening 132,000, for example, for us is not easy. So can AI help in this aspect? So this is a hypothesis that I made, and then luckily we got funding from google.org for this one, and then using that we have developed a AI-based methodology which massively increases the throughput of screening. So this is one example here. We have a hypothesis that the whole collection of 132,000 can be screened for a trait of interest in one season. But as a pilot, we only took up 60,000, which is about half of the collection. And did we do it? So flooding, many of you in the rice business know flooding is an important trait, and a gene such as SUB1 was used and solved the problem. But because of climate change, now SUB1 can give 14 days of tolerance. now it is not sufficient. Farmers are demanding 21 days of flood tolerance. So how do you do it? So we go to the gene bank, search it, do it, tedious. So with this AI-based approach, we were able to screen 60,000 collection, of almost half of the collection for submergence tolerant and identified donors here. So this is an example, just photo of how the trial looked. This is just concluded 2 months ago in the IRRI nursery. So in summary, as I explained, ever since IRRI Gene Bank started, since 1977, till 2023, only 20,000 accessions were ever screened, and sub-1 came out from that one. But in 2024 dry season alone, we screened 60,000, and we identified 1,200 tolerant accessions. So this is a very rapid way of screening the collection and then identifying the used ones, and then spend more time characterizing the better ones. So we estimate cost is 1/10 of the traditional way of doing things, time, and cost is 1/16. So this is a massive benefit to the gene bank to screen large collection. And this can easily be extended to any other seed crops. And within rice, we are now targeting the biotic stresses going forward. And similarly, we have also done for drought. For drought, nowadays, IRRI used to work a lot on the reproductive stage, but now seedling stage is also becoming important. So, and seedling stage both in the upland condition as well as the lowland conditions, and this is an example of how we have been able to screen 60,000 for this stress as well. Similarly, for salinity, and this was done at a EC of 18, which is quite high, and as you can see, there are so many good lines, as good as the Czech, or even better than the Czech in the collection. And so with the same number of people, only using 5 people, we were able to screen 60,000 for this. So this is a massive improvement over the traditional way. So now again, I want to switch gears and talk about something different. All the while I was talking to you about how we can support the IRRI breeding strategy, which I showcased in the first slide, right? So how we can support that. Now, I want to switch gear. Can we do something more than that? So it is, one of my hypotheses is breeding programs have generally been built using the locally available germplasm. It is not based on the best global germplasm out there, which is a limitation, according to me. So what we did, we chose our partners in Vietnam, and we introduced over 4,500 exotic germplasm from all the regions outside of Asia. We didn't want to put Asia. We took material coming outside of Asia and Africa because of the numbers, and we introduced them into Vietnam and did testing over a period of 3 years. This is the last year that we are going to see, and we have very interesting results. So yeah, this is the slide which showcases all the testing, screening that we did together, and I'm calling it as participatory access and selection, and it is very similar to participatory varietal selection, where you do the selection for the breeding material. So such selection can also be done for the gene bank material. Right? So this is the 3rd year, and we have results from 2 years already, and the results of 1st year and 2nd year is highly consistent. I only present the results of the 2nd year. So we have 2 locations, Hanoi and Mekong Delta. So here you can see, surprisingly, some of the material are even doing better than the red bars are the local checks, the best local checks the farmers are growing, recommended by the national program. Blue ones are the gene bank material. A lot of gene bank materials are doing better than the best checks. Okay? And as you can see on the right-hand slide, nothing from Australia was good. Nothing from Europe was good. Nothing from Central America was good, but mostly South America. Somehow, they like Vietnam. So they're doing well. So, and the results, when you go to Mekong Delta, are even more striking. You see, the lines coming from gene bank material from South America are doing almost 50% better yield than the best check of that region, which is, I didn't expect this result myself. And our partners are so impressed with that material, with this, they want to release these materials in Mekong Delta. These are gene bank materials, and they want to release them for direct cultivation as it is, so because the effects are so large. And again, so see the situation, nothing from Australia, Europe, is doing well, but only from South America. So this is what I call as G by E. So when I show this slide, many people ask, why do we need breeding? We just introduced this one, right? I am a plant breeder myself, but what I am saying is, we should use this as the starting material to breed. Then you will have high impact. So this is a way to support the whatever approaches whatever we want to do, much more than just supporting the existing breeding strategy. So just a snapshot of what are this material. Some of this material entered into the gene bank in 1963. More than 40 years they were sitting in the gene bank, and today you take it out and test out, and they are beating with the best checks. So what is the underlying reason? Maybe the climate change. Today the climate situation in Vietnam is far distant from what it was 30, 40 years ago, and now today better set of material are suited to you. So instead of beating a small, their own breeding lines to increase 1%, 2%, there is more drastic way of improving the yield for that region. So this is what we have found, and I think this is interesting, and we want to try this even in other regions. So those were some of the things that I wanted to highlight in my talk, and again, I would like to thank the organizers for giving me this opportunity. And this is my team in IRRI in Philippines, and these are the collaborators. Some of them are here in this room, and I would like to thank all of them for their contributions. Thank you so much.
Thank you, Dr. Ramaiah, to let us know the outline of IRRI strategic approaches, and then let us learn how important the role of gene bank to harness genetic resources. So thanks so much. Once again, please give him a round of applause. So now, please let me introduce Dr. Yoshihiro Matsuoka, Professor, Graduate School of Agricultural Science, Kobe University. His current research interests include investigating the adaptive mechanisms in wild wheat, exploring the genetic and the ecological nature of the origin of common wheat, and finding ways to better utilize wheat genetic resources in breeding. Prof. Matsuoka, please start your presentation.
Okay, thank you, Chair, for introducing me, and many thanks for the organizers to inviting me to this great conference. So let me say something funny, and my name is Yoshi. Okay, so my name is Yoshi. So I'm from Kobe University, and then this green monster is also Yoshi, called Yoshi. and of course, this green monster is not from Kobe University. This green monster is from Nintendo, as you might know. So I think I'm lucky to share the same name with this green monster, and because this green monster is world famous. But I'm not sure, but I'm absolutely not wide famous. So anyway, I just wanted to be funny. And then let me start. And then thank you very much for coming today. And then I hope you'll enjoy my talk. So now the climate is changing. And the climate change will have a strong impact on our food production. And the bread wheat production is no exception. And so this slide shows an example of such impact. And there we see stimulus disease is predicted to expand significantly in Europe in 2050 under the predicted climate conditions. So other than diseases, so there are many things could happen. And so looking at what is happening right now and looking at what is going to happen in the near future, it is almost clear that we need to improve bread wheat and we need to make it better adapted to the future conditions. So one thing we could do to improve bread wheat is to use wild wheat species as genetic materials that can be used in breeding. So today, I think I'm going to talk about wild wheat species named Aegilops tauschii. So here are the spikes of Aegilops tauschii. And then I wanted to show this photo in the beginning of my talk because I thought some of you might not have seen actual plant of this species. So as you can see, the spikes are diverse in shape, with some having straight outlines and others having curved outlines, along with a variety of intermediate forms. It is always actually fun to me to observe the spikes of this species, and I'm sure you'd enjoy the diversity when you see the actual plants in person. So let me start by explaining why Aegilops tauschii is important as a genetic resource in the field of wheat breeding and genetics. So as is well known, Aegilops tauschii is the father to bread wheat, Triticum aestivum aestivum. And it was about 8,000 years ago that the species hybridized with Triticum turgidum, the mother to bread wheat, and gave rise to interspecific hybrids, the direct ancestors of bread wheat. So one reason why Aegilops tauschii is important lies in the fact that the species hybridized with Triticum turgidum multiple times. But it wasn't like the hybridization took place many times at many places. So let me explain this using the map on the right, and there you see the distribution of Aegilops tauschii in blue, and apparently the species has very wide geographic distribution across the Eurasia, a central part of Eurasia from Syria to China. And in the red region, the species hybridized with Triticum turgidum. But outside the red region, species never hybridize with Triticum turgidum, even though the species is widely spread outside the red region. So the essence of the matter is shown in the figure bottom right, and there we see bread wheat inherited only a small portion of Aegilops tauschii alleles due to the limited number of hybridizations. And the alleles not involved in the hybridizations were not inherited to bread wheat, and they just remained within the species. So in this sense, Aegilops tauschii is a huge reservoir of alleles that may not exist in bread wheat. And such alleles may confer useful phenotypes when transferred to bread wheat. So this is one reason why Aegilops tauschii is important. And I think we'd like to explore this huge reservoir for use in breeding. So now let's say we have accession, Aegilops tauschii accession, showing a useful phenotype. And then how can we transfer the alleles controlling the phenotype to bread wheat? So luckily, we have a relatively simple method. That is, we can cross this, our favorite Aegilops tauschii accession with Triticum turgidum and produce a synthetic wheat carrying the alleles we want to transfer. And then this synthetic wheat is genetically equivalent to bread wheat. So we can cross this synthetic wheat with bread wheat variety and then transfer the Aegilops tauschii alleles to the variety. So this way we can transfer Aegilops tauschii alleles to bread wheat by crossing. And then maybe we can transfer Aegilops tauschii alleles to bread wheat maybe within a couple of planting seasons, but usually further back crossing is necessary to produce a practical commercial variety. So now we have a collection of Aegilops tauschii accessions, and we have a method to transfer alleles to bread wheat. But still, there is one other thing which is nice for us to have. That is a map of genetic diversity that guides us to accessions which we might want to use in breeding. So here is an example of that kind of maps. And in this map, we see the structure of Aegilops tauschii germplasm, which consists of 3 lineages named tauL1, tauL2, and tauL3, where our lineage is defined as a group of accessions that are genetically similar to each other. And in this map of genetic diversity, we also see tauL3 and tauL2 are relatively close to the digenome of bread wheat, whereas tauL1 is kind of distant from it. So this genetic map and this map of genetic diversity is useful when selecting accessions for testing. For example, tauL1 is a great option if we aim to discover alleles that are significantly different from those in bread wheat. So the lineages, these lineages have distinctive natural geographic patterns. So here is a map of lineage distribution, and there we see tau L1 is widely spread across the species range, whereas tau L2 and tau L3 are limited to the western part of the species range. So this information is important, and then so probably in the ancient times, tau L1 was the driver of species range expansion, where from the western part of the species range, where all three lineages existed, to the eastern part, where at the time of range expansion, Aegilops tauschii wasn't present. So this information and this observations is important because it shows tau L1 is the lineage that adapts to the Central Asian environment, whereas tau L2 and tau L3 do not. So I think tau L1 is an interesting lineage and important lineage. So I will talk more about this lineage later on. So let me first talk about tau L2 and tau L3. So as we can see in the plot, tau L2 and tau L3 are relatively close to the region of bread wheat. So it is reasonable to ask if whether they are crossable with the Triticum turgidum, the mother to bread wheat. So we performed a cross experiment using 85 accessions of Aegilops tauschii, and the results and the tester was a single variety of Triticum turgidum, and the results are shown in the figure bottom left, and there we see tau L2 and tau L3 were actually much crossable with the tester relative to tau L1. So also we observed that some tau L2 accessions were exceptionally highly crossable with the tester. And also we see in the map, these highly crossable accessions were all from Iran, Azerbaijan, and Georgia, the cradle region for the evolution of bread wheat. So based on these findings, we can infer that the ancient Aegilops tauschii that grew in the cradle region and eventually became ancestral to the region of bread wheat probably had high crossability with Triticum turgidum. And this high crossability probably contributed to the formation of natural hybrids between Triticum turgidum and Aegilops tauschii about 8,000 years ago. So another thing I would like to report on this occasion is the discovery of tau L3 in Iran and Azerbaijan. Previously, tau L3 was known from Georgia only. However, we recently discovered 3 accessions from northern Iran and 2 accessions from Azerbaijan. So probably this lineage has wider geographic distribution than previously thought. And also, this information is important because tau L3 was involved in the formation of the region of bread wheat. So these newly found accessions will enable us to study the role of this lineage in the evolution of bread wheat. So also this discovery is informative to our conservation activities because tau L3 is a lineage of a very small population size. So I think we need to take care of this lineage and for this lineage not to die out. So now we must extend our conservation activities to Iran and Azerbaijan outside Georgia. So, okay, now moving back to tau L1, and I think I'm going to talk about why the lineages can be informative and how the lineages can be informative, studying agronomic traits by taking the case of salt tolerance. So in this slide, we see 3 Aegilops tauschii accessions undergoing germination and seedling growth. And then we see accession C can germinate and grow even at 1.5% NaCl, whereas accession A cannot germinate and grow at 1.0% and higher. And accession B cannot can germinate and grow at 1%, but cannot grow at 1.5%. So the Aegilops tauschii accessions were variable in the degrees of their salt tolerance during germination and seedling growth. And of course, we'd like to find more accessions like accession C for use in breeding for salt tolerance. So we examined the natural variation patterns for salt tolerance during germination at 1.5% NaCl using about 200 accessions. And we found 54 accessions in which at least 4 out of 9 seeds germinated. And we considered them to have salt tolerance during germination. And we also found these salt tolerant accessions were widely spread across the species range, as we can see in the map. So we didn't observe any apparent structure in the natural variation pattern in terms of geography. However, when we observed the natural variation pattern using lineages, a clear structure emerged. That is, the accessions having salt tolerance were all from tau-L1. So the natural variation pattern for salt tolerance during their germination was actually structured in terms of geography. So clearly, lineages can be informative when, if we aim to discover salt tolerant alleles, and of course, tau-L1 is the lineage we might want to prioritize when screening accessions. So the lineages can be useful in exploring Aegilops tauschii germplasm, but I think I must point out the downside. So the downside, I think, is the lineages are kind of difficult to distinguish based on morphology. So we need to genotype an accession to figure out which lineage it belongs to. So let me explain the situation using the spikes. So in this slide, we can see the spikes of Aegilops tauschii, and the first 4 are from tau-L1, and the second 4 are from tau-L2, and the last 3 are from tau-L3. So as we can see, the spike like this one is kind of easy to tell because it has very curvy outline, so it is easy to tell this one is from tau-L2. Otherwise, it is kind of difficult to tell exactly which spike is from which lineage. So it would be very convenient if we could tell the lineages just by looking at the spike shape and without doing genotyping. So in order to address this issue, we took a machine learning approach, and then we fed thousands of spike photos to a computer and had it find a way to classify the image, referring to the lineages as lineages used as the labels. So this is an example of a computer view of a spike image. And as we can see, in this case, computer was looking at the spikelet shape as highlighted by red in this figure, and then to classify this image as tau-L1. So in this case, the computer was making sense because spike shape is a key trait to classify subspecies of Aegilops tauschii. So we think, we felt this approach might work, and then we are working on adjusting parameters, and then I hope we can report the final result in the near future, somewhere in the near future. So here is the conclusion. So I had 2 points in my talk. And then the lineage, the first one is the lineages are useful in exploring Aegilops tauschii germplasm. And the second point was tau-L1 is an important genetic resource for improving bread wheat. So I'd like to say thank you very much to these persons for collaborating with me for a long time. And also I'd like to say thank you very much to the Kakenhi grant founders. So this is, I think, the end of my talk. Thank you very much for your attention.
Thank you, Prof. Matsuoka. Yeah, we know wheat is very important and to major crop. And thank you so much for letting us understand the potential of wild wheat for developing new variety. Once again, please give him a round of applause. So last but not least, we'd like to call Dr. Shinsuke Yamanaka, Director of Tropical Agriculture Research Front, and then project leader, JIRCAS. His area of expertise is studies on plant genetics resources continually from his graduate study. and currently conducting research on the diversity and the utilization of tropical crop genetic resources, including tropical food. Dr. Yamanaka will share with us JIRCAS tropical agriculture research front engagement in genetic resource studies. So Dr. Yamanaka, floor is yours.
Thank you very much for introduction, Dr. Hayashi. And first of all, thank you very much for giving me this opportunity for organization committee. Today, I would like to introduce our research activities on tropical crop genetic resources in JIRCAS. So I'm from the Tropical Agriculture Research Front, TAF. And let me introduce our research station, TAF. TAF is one of the research divisions in JIRCAS, which is only located outside of the Tsukuba headquarters campus. So we are in Ishigaki Island, Okinawa Prefecture. And the location of TAF is, the latitude is 24 degrees 22 seconds, longitude is 124 degrees 11 minutes east, and subtropical climate and island environment. We have around 30 hectares campus, including 21 hectares of experimental research fields where research is conducted on a variety of tropical crops. Facilities include greenhouses, research fields, and open laboratories, as shown here. One of the advantages of TAF is the tropical crop can be grown outside in the field, namely in actual growing environment for experimental research. Taking advantage of this geographical advantage, we promote JIRCAS project research and also conduct activities that contribute to agriculture in Japan, especially for Nansei, Southwest Islands. Now, so I move to the project introduction. Tropical Crop Genetic Resources Project. Actually, this project is located under the information program led by Dr. Iyama of JIRCAS. It means we are conducting not only breeding and cultivation researches. This project is also collaborating with other projects in JIRCAS. The other projects involved are indicated by red boxes here. And JIRCAS has a large number of diverse genetic resources of tropical crops, sugarcane, indica rice, tropical fruits, and urokuroa, tropical grasses, as shown here at TAF. These tropical crops play an important role as food, energy production, calorie, and nutrient sources, cash crops, and animal feed, forage in production areas. We are also developing technologies to utilize these genetic resources. This project is designed to utilize these genetic resources and technologies at TAF to conduct researches on genetic resources to contribute to JIRCAS mission. Taking these backgrounds into account, the project aims to develop varieties and technologies that take advantage of the diversity of genetic resources by sharing issues with international and domestic collaborating research organizations. This project emphasizes external collaboration such as network formation and sharing of information, technologies, and materials. I will now explain the project themes. There are 4 themes. First one, information and networking of genetic resources. Focus on the diversity information for advanced utilization of genetic resources and sharing information, technologies, and materials by research networking. Second one, evaluation of genetic resources and development tools involves the development of evaluation method and tools for agricultural characteristics and environmental stress tolerance, genetic analysis of useful traits, and functional analysis of useful genes. Third one, utilization of genetic resources for new breeding materials and cultivation technologies. We are developing varieties and breeding materials for unstable environments and developing cultural practice techniques based on the variety and the environment. The last one, domestic research collaboration through utilization of genetic resources. It focuses on the development of the materials and the technologies for domestic needs, variety diversification, stable production, cooperation for measures of global warming and breeding program in Japan, and external cooperation through providing materials and technologies. Next, an overview of research activities provided. The actual research activities can be roughly divided into two. One is strengthening of genetics resources utilization, shown in blue, and second one is advancement of genetic resources utilization through external collaboration, green one. For the background of the project, the first issue to be addressed is the response to climate change. In our target crops, there are high temperature during flowering and drought in various crops, irregular weather during the rice heading period, and reduced production of mangoes due to high temperature during the flower bud formation period. In response to these issues, development of varieties and cultivation techniques that can mitigate the effect of climate change and global warming is required. And stable production in adverse environment is also a major problem. Salinity, acidic soils, and diseases. Tolerance to these biotic and abiotic stress conditions and cultivation method and harmonization with the environment are necessary. As for genetic resources themselves, there are many that have not received much attention or have not yet been unutilized. In the case of rice, there are cases where breeding and utilization has not progressed due to the reproductive barrier between related species, for example, between Asian rice and African rice. Evaluation of native cultivars that have gone unused as a result of majority production of a few major varieties, as in the case of mango, and in sugarcane, where the genetic background is existing varieties narrow, and the genetic background of closely related and wild relatives has not progressed. So, as an example of our activities, we present the improvement of the root using the relatives of sugarcane, Elianthus, in an intergenic hybrid produced to add the long and numerous root characteristics of Elianthus to existing shallow-rooted sugarcane. The roots extend deeper into the ground than in existing sugarcane variety and may utilize water deeper into the ground, even under drought conditions. In the case of rice-related species, that are difficult to hybridize due to reproductive barrier mentioned before. It was found that the diploid F1 has almost zero pollen fatality, while the double haploid line through tetraploid recovered fatality and produced seeds as shown here. It becomes a breakthrough for the development of efficient breeding material methods. As for evaluation of diverse genetic resources, we show the example of the evaluation of sugarcane and mango characteristics under stress conditions. In sugarcane, the water use efficiency index was used to evaluate drought tolerance, and variation among varieties was found. In mango, we investigate the temperature at the half reduction of photosynthesis under high temperature conditions and investigate the relationship with stomatal response. Genetic resources showing high performance in both cases are expected to useful materials for breeding stress-tolerant varieties. As for development of breeding materials, a group of rice lines with various heading-related genes were created, and the actual heading dates were evaluated. The red and blue colored lines in the graph show statistically significantly longer or shorter days than the reference variety IR64. The dark red and blue ones show various variations that extend the range of existing genetic resources. The lower figure shows the result of the evaluating the effect of accumulation of resistance genes, gene pyramiding in different types of the rice blast. Those in blue are combinations of lines and strains showing resistance. We are also in the process of evaluating plants through actual cultivation. Development of new varieties and evaluation of breeding lines on site are also progressing. The variety KK4, Konken4, developed by JIRCAS in collaboration with Thailand. This variety was adopted as recommended variety by the Thai government last year. It is expected to grow better and produce higher yield than the existing variety KK3. Isan, a Urokuroa tropical grass variety developed by JIRCAS and registered as a variety in Thailand just last month. It is expected to contribute to the stable production and supply of tropical pasture grasses. The lower image shows a trial of an improved line of Ciherang, the main variety in Indonesia, for AWD cultivation. So genetic resources to information resources, namely accumulation, integration, and sharing of genetic resources information, we have conducted and released a database of characteristics information on the Eliansas and mango. We also plan to release information on wild sugarcane species and tropical rice in the future. As activities related to networking with production countries and areas in each crop, we are working to strengthen collaboration by promoting information sharing and provision with organizations in each country for sugarcane, tropical fruits, and rice. Collaborations for domestic agricultural research are cooperation in national sugarcane breeding programs, development of virus-free technology for seedlings as a countermeasure against the passion fruit virus disease, which was spreading seriously in Okinawa recently, and functional analysis of genes for early morning flowering rice plant using transformation and gene editing technology to analyze, conducted in collaboration with other JIRCAS projects and other domestic institutions. So this is a summary slide. In this project, we are working on genetic resources diversity, technical basis, and application and implementation, as well as promoting project activities with a strong concern for center function through networking, information sharing, and research collaboration activities in order to advance genetic resources utilization. We will continue our effort to ensure that these initiatives contribute to stable production, genetic resources utilization, and food diversification. Finally, the implementation of this project is being carried out in cooperation and collaboration with the international domestic organizations mentioned here. I would like to conclude by expressing my sincere gratitude to each of these organizations and express my hope for further collaboration in the future. Thank you very much for your attention.
So thank you so much, Dr. Yamanaka. So it's the end of session, and before closing this session, please give one another time your round of applause to all presenters.